Chemiosmosis produces ATP in both chloroplasts and mitochondria using the same mechanisms. In both cases, an electron transport chain maintains the hydrogen ion gradient that powers chemiosmosis. There are differences, however. The electron transport chain in the mitochondria and the chloroplast differ in their ultimate source of energy, their electron donor, their electron acceptor, and the regions of high and low hydrogen ion concentration. Let's look at these in a little bit more detail, starting with the mitochondrion. Here is the electron transport chain in the mitochondrion. The electron transport chain is a series of protein complexes embedded in the inner membrane of the mitochondrion. Electrons are harvested from glucose and other food molecules, and then these high-energy electrons are shuttled to the electron transport chain by NADH and FADH2. These are the reduced form of the electron carriers of cellular respiration. So NADH and FADH2 are the electron donors for the electron transport chain in the mitochondrion. Electrons are passed from molecule to molecule, and this occurs because each component of the electron transport chain has a higher electronegativity than the one before. Recall that the electronegativity is the ability of an atom or molecule to attract electrons. So electrons are passed from molecule to molecule. You can see them being passed from complex 1 to 2 to 3 to cytochrome C to 4. And each of these redox reactions releases energy. Some of that energy is used to pump hydrogen ions from the mitochondrial matrix, which has a low concentration of hydrogen ions, to the intermembrane space, which has a high concentration of hydrogen ions. Because we are transporting something from an area of low concentration to an area of high concentration against its gradient, this is active transport. The electrons are finally passed from complex 4 to oxygen. Oxygen is the final electron acceptor of the electron transport chain in the mitochondrion. And recall from all the way back when we talked about chemistry that oxygen has one of the highest electronegativities of all the atoms on the periodic table. 2 electrons combine with an oxygen atom, as well as 2 hydrogen ions, and form water, which is a product of cellular respiration. This right here is the reason that we need to breathe oxygen. Without oxygen, the electron transport chain would no longer work, and then the concentration gradient of hydrogen ions between the inner membrane space and the matrix would be equalized, which means that ATP synthase would no longer function, and the bulk of ATP production in the cell would stop. There's also an electron transport chain in the chloroplast. This one functions a little differently, but the overall punchline is the same. Hydrogen ions are transported against their gradient so that they can diffuse through ATP synthase, producing ATP. In this case, the source of energy is light. Light energy excites electrons in a large complex called photosystem 2. Electrons are then passed down an electron transport chain that is embedded in the thylakoid membranes of the chloroplast to photosystem 1, and then ultimately to NADP+, which becomes reduced to NADPH. Some of the energy released by the exergonic process of electron transfer is used to pump hydrogen ions from the stroma into the thylakoid lumen. Once again, this is active transport because we are transporting something against its gradient. These hydrogen ions can then diffuse or flow down their gradient through ATP synthase, powering the production of ATP. This is how light energy is captured and transformed into the chemical energy of ATP and NADPH during the light-dependent reactions of photosynthesis.